Okey, seterusnya saya ingin menjemput penceramah jemputan yang terakhir untuk hari ini, iaitu Profesor Madya Dr. Ramani Mayappan yang akan berkongsi tentang penulisan, result dan discussion. Sebelum itu, izinkan saya berkongsi bila teringkas berkaitan beliau. Profesor Madya Dr. Ramani Mayappan merupakan pensyarah kanan sains gunaan dari UiTM Cawangan Perlis. Beliau memperolehi PhD dari Universiti Sains Malaysia dan bidang pendidikan beliau ialah intermediate. Beliau telah dilantik sebagai ketua editor untuk jurnal Intellect dan yang terbaru untuk Scientific Research Journal. Beliau juga aktif sebagai pengulas ataupun reviewer untuk beberapa jurnal seperti Journal of Alloys and Compounds. Beliau juga telah menerbit lebih dari 39 terbitan termasuk jurnal Bioinfak dan Proceeding. Hadirin sekalian, tanpa melengahkan masa, menjemput Profesor Madya Dr. Ramani untuk menyampaikan perkongsian ilmu beliau. Dipersilakan, Doktor.
Okay, can you hear me?
Yes, Doktor.
Okay, thank you, Dr. Rozila.
Welcome, Doktor.
Okay, today is our last session and the results and discussion. I'm also discussing a little bit on conclusion. As Dr. Zaidi mentioned just now, method is the first thing you should write, but after method, you should go for results, and after that, you have to go for your discussion. And for your information, each study is unique. So there is no one size fits all approach when you come to designing your result and discussion. Okay. So for our aim here to convert a FYP to a thesis, FYP thesis to a research paper is not a straightforward assignment. It's a lot of hard work involved. Okay. So what do you do before you start? And knowingly, as you know, doing an experiment is not very difficult. But when it comes to writing a discussion, it's very challenging, time-consuming, and also very boring work to do. Sometimes when you want to construct just one sentence, sometimes takes you hours. Okay, but the best thing to do, best approach is before you start your writing, you better set your target journal. Okay, so when you have the target journal with you, you know the formatting, how the result should be written, how the graph or figure should be drawn, and so on. So this will save you a lot of time before you submit your paper. Okay. And when you go for the target journal, okay, when you go for your target journal, you have to go through the guide for author section. Okay, this section will tell you a lot of information about the journal, and you must strictly follow the guidelines. If not, your paper will be rejected. For example, know the length limitation and restriction on the content. For example, if you go for materials letters, they are strictly only a 4-page paper. Okay, and they only allow you to submit 3 figures and 1 table, and the total words is only 2,000 words. Okay, so they are very strict on that. So it's best thing to identify your paper. Identify your journal before you start writing your manuscripts. And another thing you have to bear in mind is that some journals, they ask you to write the results and discussion at the same time, or some ask you to write the result first and discussion later. So that's why you have to clarify before you start writing your paper. And the best thing to do is you better read through the journal which the paper with the similar field was published. So the guidelines will outline specific requirement for the result section. So the published articles will provide an example of the successful approach. So this will save you a lot of time. So make sure you follow the guidelines given by the journal from your published area. Okay, one thing we have to ask ourselves, is a research done by one FYP student enough for you to submit for a journal? And I don't think so. So normally what we do is we combine results from 2 or 3 FYP students, and by combining them, in fact, while they are doing the experiments, we have to monitor them carefully so that the method is followed strictly for guidance. And also, after you got the results, okay, basically you have to rewrite everything: the abstract, the intro, the method, the result and discussion, the conclusion and references, because you are combining 2 or 3 theses, so you have to rewrite everything, and make sure it's not too long or too short. Make sure it's around 10 to 15 pages. And bear in mind, when students start writing the thesis, they barely have time to write the thesis. They will spend around 2 to 3 weeks before the submission. They will be very careless in reporting, especially the discussion and citations section. So when you rewrite, you have to read the manuscript, you have to recheck the citation and make sure the citations are correct. Okay, when you talk about results, what's included in the results? As Dr. Zaidi mentioned just now, you must follow the order, okay? Make sure the results are relevant to your objective, okay? And explain the least important to the most important. For example, when you characterize, for example, you report the melting temperature first, density, if you have FTIR or SEM or XRD or BT and so on. Then you go to the more difficulty, for example, aging or sintering or NMR or TEM and so on. If your scope is broad and has many variables, the author should select results that are most relevant to the objective. Okay, so you have to be very careful in that. Don't let the reviewer points out your mistakes. Okay, when it comes to data presentations, we are quite familiar with figures, tables, graph, and semantics. So the difficult thing here is we are combining data from 2 or 3 FYP students. So basically, you have to combine the tables, charts, graph, and everything. You have to combine or redraw or replot the graphs. And come to this, you have to analyze carefully. And make sure all the figures, it should be standalone. That means when you have a figure, the reader or the reviewer should be able to understand your figure or tables without reading the text. And of course, as Dr. Daisy mentioned just now, we were talking about high-end instruments. Nowadays, results from SEM or FTIR are very common. Researchers are going for high-quality display. And they say we must give our work a professional appearance. For example, this is what they call SEM. They call it mapping. Okay, and this is also quite costly. So each element gives you different color base. So if you can produce this kind of diagrams, It gives you professional appearance. Another example is this one. We're talking about carbon nanotube. And also, this is TEM, and the number D here is, this is a TEM, okay, showing the carbon nanotube. And this figure here is, the other one is, the bottom one is actually from my area. When I did my PhD, just use SEM. But now this graph is based on Raman's spectroscopy. At that time, they were not using this instrument, but now they're using it. So you can see how much changes have occurred. Okay, another good example is this one. This is a result from TEM for solder material. And the interesting thing here is the TEM is done, what do they call it, in situ testing, whereby the heating, this is heating and isothermal and cooling was done inside the TEM. And the TEM diffraction was captured. Okay, don't be surprised. This is done by our neighbor, our UNIMAP student. Okay, she's not a student, but she's a postdoc student. I think she was doing in Australia, and this TEM was done in Japan, I think, if I'm mistaken. So when you have a very professional-looking monostrips, the reviewers might think your results are very good and your interpretation is reliable. That's a very good thing about having a high-end instrument. Okay. As I mentioned just now, make sure your figures, tables, and graphs stand alone. No? So you should be able to explain everything. So if you have good results, make sure your figure tables are impressive. So when you talk about impressive, this is very impressive. This is, as I mentioned just now, this is done by the UNIMAP staff, whereby she has 3 different solders, three different solders at 4 different temperatures. So you plotted everything in one figure. The diffraction was captured during melting in the TEM. Okay, so this is very impressive, and she published it in this journal, Material, and she won an award for this uptime material group of journals. This little girl, Flora Somidin from UNIMAP. I think she's doing postdoc in Australia. So this is very good actually. So when you have results, which results you should present? So figures should supplement text, the text, and not just repeat what you have already mentioned in the text. Because readers might rather see a graph rather than that, because we are very busy normally. When you go through papers, we just refer to the graphs and tables. We don't read the text. Okay, when you talk about figures, for example, this SEM here, some journals might require you to what you call redraw. This is the SEM, complete SEM, and this 5 micron is what you call the error bar. Sorry, the MAC here, the scale bar. Some journals, they ask you to redraw the scale bar. So we have to we have to crop the picture and redraw the scale bar so that it's clearer. If you compare to this diagram here, where the scale bar is not very clear. So make sure your scale bar is very clear. Okay. And this is SEM. But if you use optical microscope, basically there's no scale bar in optical microscope, and sometimes the supplier don't supply you. Actually, for optical microscope, the scale bar is sold separately. There's a glass box. A glass box which has the scale bar. So you have to snap the picture and also the scale bar separately and that combined. And when you talk about magnification using optical microscope, you have to bear in mind microscope has two lenses. One is eyepiece and also the other one is objective lens. So when you come to magnification, you have to times them. So you make sure you don't make such mistakes when we do, especially when the students they don't realize that you have to tell them about this. Okay, when you talk about graph, okay, of course, when you draw a graph, of course, when you talk about FYP results, when you, as I mentioned just now, when you combine 2 or 3 FYP results, you have to combine the results, and that's when it's consume time, and you have to really redraw the graph. Make sure you label the axis, specify the units and for the quantities, label all curves and data sets, and make sure the font size is readable, not too small. If you make it too small, then the editor might ask you to redo it again. Okay, so this one you have to graph. And as you know, graph is basically to show a relationship between 2 or more items. Okay, whether it increases or decreases or no changes. So details about individual data points we're not bothered. So that's what you call advantage of having a graph. We don't bother about data points. So when you combine the graph, for example, this one is DAC results from 3 different students. I combine them in one graph. This one I think I use Excel. Okay, so you can see what you call changes in melting temperature. After graph is the other one is this is another combined graph from 3 students. The tensile test using tensile instrument. By you combine 3 graphs here. So you plot stress versus strain. Okay, another one is semantics. Okay, semantics basically to identify key parts of a system or process that cannot be highlighted in the text. If we have difficulty explaining in the text, we draw schematic diagrams. For example, this one is diagram A shows what you call diffusions of tin and copper atoms, and the other one shows how carbon nanotubes retard the diffusions of SNN and also copper atoms. So after adding the carbon nanotube, you can see the intermetal thickness decreases. So this is this graph explains quite clearly how it was done. And this was actually this was done by FYP student actually. Okay, I'll show you later. This is done by our FYP student. And for more complicated semantic diagrams, you can have this. Okay, this I think the author must have spent a lot of time in drawing this diagram. This one shows how they mix solder powders with carbon, what you call graphite nanotubes, how the mechanism works. It's shown step by step. And this one is this one shows how what you call nanotubes could be used to harvest energy from a fluid. This is from research, nano research journal. Okay, when it comes to image editing, first thing you have to remember, you must avoid image manipulation. Don't try to manipulate your image because when you manipulate the image, you might what do you call try to avoid what do you call bad things. You try to just display the good things, that is wrong. And another one is adjusting the contrast. For example, these two diagrams are same diagrams, so it's different contrast. The first, okay, this is SCM diagram. As we know, SCM diagrams this is the best scattered image of SCM diagrams in metallic actually. We must remember how SCM works actually. SCM backscatter works in a process whereby higher atomic numbers give you brighter image, lower atomic number give you darker image. So that's the contrast there. So if you adjust the contrast, for example, the second one, when you adjust the contrast, it becomes brighter. You can see all the scratches there, so which doesn't look very nice actually. The first one looked better with less sketches. But the second one, the first one gives you a lot of dark spots here. That shows a very bad polishing. So here you must really be careful how you select your SCM diagrams. And also image cropping, I think as I mentioned just now. And if you use any specific software, you have to state the software that you used. And whatever you did for your image, make sure you keep the original images and files. And also they call it metadata. Metadata is basically, if let's say you have a data, all the information regarding the data, they call it metadata. You have to keep the metadata. Sometimes the editors might ask for the information. Okay. Okay, figure quality. Figure quality is very, very important. For example, this is an example. This is a FTIR image sent to General Intellect when I was the editor for General Intellect. And one of our staff actually sent this image for publications. So I have no choice but to reject that paper because I cannot see the lines. The lines are very blurred. Okay, so you have to be very careful when you submit your images. This is a very pity actually. Okay, tables. Tables is an effective way to present if you have a large amount of data. So design them carefully so that you clearly communicate your results. Okay, make sure your columns are correct, concise, legends are properly written, captions are given. Remember, for captions, table is written on the top of the table. For figures, they are written below the figure. And the data are divided into categories for clarity and sufficient spacing between columns and rows. And also when you write units, make sure there's a space between the numbers and also the units. There must be a space there. And the font size is readable. And make sure your table is in editable form. Don't send as an image because when it comes to publications, the publisher will edit your table. So you must remember your table should be editable. Don't send as an image. And for the format, make sure you refer to the published article in that journal so you can see how they are formatted for the table. Okay, captions for tables and figures. Since we are talking about FYP students, generally FYP students they're not very good in writing captions. For example, if it's a SCM just wrote the SCM micrograph, so it should be SCM micrograph of a sample X aged for let's say 600 hours for 1000 degrees at 1000 degrees, for example. Okay, so it has to be very specific. As I mentioned, the caption should be standalone. No? Because it is very crucial to know how to caption the figure and tables and refer them within the text. Okay, and one thing you have to remember, when you refer your tables and figures in the text, make sure you mention about them first before you paste your figure and table. We should follow after that. It's not before. And best thing to do is to check the requirements and standard of the journal to which you are submitting your work. Because every journal has its own design and layout standard. You have to have multiple, if you have multiple variables, it's better to split the table and or figures. There's a technique to write your captions actually. For example, let's say your objective is, let's say for example, to study the effect of temperature on microstructure of sample X, for example. And that your caption is microstructure evaluation of sample X at A 500, B 600, and C 700 degrees Celsius. So you follow the objective. Let's say for your objective is to study cadmium concentration in different parts of onion after 14 days, for example. Your caption should be mean concentration of cadmium determined in A leaves, B roots of onion, and after 14 days period. So your caption follows the objective. So not only that, you can see here the captions sort of stands alone. Okay, that is for results. We go for discussion. Okay, generally, when you talk about FYP students, they tend to repeat things. So make sure you don't repeat your method or introduction in your discussion. Okay. If you have large amount of data, you better plan your approach. As I mentioned just now, for example, your melting temperature, your density, your SCM, and so on. And if it is too long, make sure you can use sub-headings to improve your readability. If you have, let's say, 3.1, 3.2, and so on. And this is also depends on your journal requirements. Some journals, they don't use subsections. Some they do. And one obvious mistake by our FYP students is the tense. They don't write in past tense because they follow their FYP proposal. They will stick with will do and will do. And so you have to be careful with that. Whatever terminology and abbreviations, make sure you already explain it in your introduction section. And as I mentioned just now, writing discussion is very, very difficult. For a start, write whatever strike your mind. Just write it. Don't bother about grammar and whatever it is. Just write, and you can always edit later. Okay, when you write discussions, the results sometimes agrees with the literature, sometimes they don't agree with the literature. Okay, so if it agrees with the literature, you can always write, for example, John et al. Reported a similar results, for example. Okay, and if it doesn't agree with you, the results don't agree with the literature because, when you say because, firstly, you must remember, of course, because you must explain as best as you can, and also make sure you cite the paper that does not agree with you. Okay, because references have an important role in many parts of a manuscript. It is very, very important. Failure to sufficiently cite other work can reduce your chances of being published. Okay, so don't let the reviewer saying that you don't have enough citations to support your discussion. It just will be very bad. Okay, and also when you have a conflicting results, when you cite people's result which is conflicting with you, it's also a chance to explain why you think your results are different. So you can explain to them why your different results are different. Okay, and every statement of fact or description of previous findings requires, of course, as I mentioned, supporting references. The references should be the latest. The latest, I think, 5 years now. Attention should also be paid to journal requirements because some require a certain percentage to be between 10 to 5 years of time. Because citing the latest references is important because journal editors see that there is a potential reader or potential audience for your manuscripts. When you have cited latest references in your field, that means there's a potential audience for your paper in that area. And also make sure you cite journals from top ranking in journals. Okay, that is very, very important. And of course, not too many references. And also one important thing I have to mention here is make sure the citations are correct. Because if reviewers are quite familiar with your area, if they know the citation is wrong, they will straight away reject your paper. And knowing FYP students, they tend to make mistakes, so you don't really, sometimes really don't trust the citation made by the FYP students. Okay, one important thing is always, when it comes to citations, make sure you read the original journal paper. Do not cite. Sometimes we are only able to have the abstract of the paper. Don't try to strike references from the abstract because abstracts don't give you a full picture of the findings. And also, do not try not to cite papers from review papers. That is, I think, it won't be because it won't give you very correct information regarding the study. Okay, you must always go for the original article, and also do not construct long sentences because long sentences will make the reader boring. Okay, and if you have long sentences, the chances to make mistakes is high. And okay, similarity index. This is very important. Of course, each journal, before they proceed with the reviewer, they will do the similarity index checking, and it is lesser than 30%. It's okay, but if more than 30%, you get rejected. And remember, when you write your manuscript, sometimes we tend to copy the sentences, especially the method, from our previous published articles. So when you do that, the similarity index will be there. So you have to be careful with that. Although your method is almost same as your published article, you still have to edit the text. If not, you will be in trouble. Manuscript rejected, one of the main reasons is there's no new contributions. That is quite obvious, no new contributions. And sometimes, although the results is good, but the way we write the paper is not adequate. So it might get rejected. And of course, the similarity index is greater than 30%. Okay. When I was the editor for Journal Intellect, there are some examples here I can show you what are mistakes made by our FYP students, because our lecturers send results from the FYP students. The common comments by the reviewers are the results and discussion part is only reporting the results. This is very obvious. Students tend to report only results, they don't discuss. There's no discussion and the results are not supported by the scientific evidence. This is a comment by reviewer. And the second one is the grammar is not up to the standard. Okay. And the third one is lack of introductions. Okay. And as I mentioned just now, captions are incomplete. As I mentioned just now, SEM, micrograph, it's not complete. It should be SEM micrograph of this temperature and this time and so on. And subtitle. When it comes to subtitle, they just put that, let's say 3.1 FTIR, that's all. It should be FTIR results for blah, 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 and so on. And one common mistake, one very glaring mistake by our, I think it's a staff actually. In the method, 5 different variables were given, but in the results, only 3 variables are there. So I think the reviewer gave a very serious comment on that. So the paper was rejected by the reviewer. So this is basically the reviewer may doubt the validity of the data because you mentioned there are 5 variables, but your results only 3 variables. So something is wrong there. So it was rejected. So another manuscript is about a homestay. Homestay revisit implementation of model-based of spatial perspective in Langkawi. This is a questionnaire research by our staff, and it was rejected. The reviewer gave you a lot of comments. The main comment was, there's no objective of the work. No main objective was given. Make sure the design of the questionnaires relate to the objective. Okay. And how you identify the homestay. Okay, I think the introduction was not, the method was not very well written. So, because there are 3 categories of homestay, A, B, and C. So it was not really specified in the questionnaire. So this is another reason. This is I show this because to show you how careless. I think this is the FYP result, must be FYP results. And other comments. Results are interesting, but should be looked at more depth and summarized. Okay. Discussion is weak, too general, and lacking of penetrative insight to reflect the author's results. So this is basically how big our results and discussions are. So this shows, so when you write our discussions, it's a very serious thing, and sometimes it reflects us also. And finally, a little bit on conclusion, as Dr. Nuzila asked just now, what's the difference between abstract and conclusion? So conclusion is basically make it 1 paragraph, and please do not repeat the results in conclusion. The key points should stand out. For example, this is from material letter. The title of the paper is, basically you answer the title, influences of AG addition on SN58BI solder on SNBI copper's interface reaction. So the conclusion is very simple, just a small paragraph. In conclusion, study on interfacial microstructure and composition of the SNBI AG solder reveals that AG content in the solder can diffuse into the copper substrate during the soldering and aging processes, forming copper AG layer at the surface. The PI segregation at the SNBI AG copper joint interface were alleviated, and the aging improvement of long-term AG will not occur. So it's very simple and very straightforward, answering the title. So this is what you call an example of a conclusion. Okay, for your information, as I mentioned, I already informed you, we have our scientific research journal with us now, and the scope is material science. So those doing material science, you can submit your paper to our scientific research journal. This is an index journal. So I think call for papers is open now for what you call March publication. So you can submit your journal for this paper. And finally, writing a journal paper is hard work. It's not what you call a misegera. You can have it in 2 minutes. Thank you.